Hi, now let's discuss about cellular principles in BME, especially cell cycle and proliferation. So the cells undergo a cycle for proliferation, and controlling cell cycle means there are many factors which controls these cell growth and division. So from a single cell to its progeny, two cells. So how do the cells know when to initiate DNA synthesis and mitosis, meaning that cells divide into two identical cells? So that decision making can come from inside or outside. So let's see this inside. The cellular protein, as there are a biological clock protein, that is a special protein to promote or inhibit the cell cycle. So this was discovered, a protein amount is going ups and downs and connected with the cell cycle. And so people named it as a kind of from cycle called as a cyclin protein. So there's even enzyme, the cyclin-dependent kinase or CDK enzymes exist inside the cell. And that decision also coming from outside. For example, if there's a cells, let's say breast cancer cells, let's say, and if the cell is getting a growth factor signal from the outside, such as here EGF or VEGF, that will signal trigger the cell to proliferate and divide. So let's look at this figure again. So if the ligand, that's primary messenger, if it happens to be like EGF, the name stands for epidermal growth factor, or VEGF, very famous growth factor called vascular endothelial growth factor, responsible for many angiogenesis process. So these factors bind to receptor that will trigger signal to this receptor, and that will goes down inside the cell, sending out that signal. You see many arrows, which means signaling cascade, meaning that that's not only one, multiple layers of happenings with a signal transduction, typically using kinase. And kinase is a phosphorylating enzyme. And usually phosphorylation means those enzymes will be activated. And so it is also coming down either cellular response or getting into the nucleus to change in gene expression, such as DNA replication. So this outside of the, the cells are constantly watching, monitoring what's happening outside. Also those getting the signal to decide whether the cells typically in a stable status and while it starts to proliferate. So there's a very interesting enzymes and parts of the DNA or at the chromosome. At the end of the chromosome, there's a repetitive DNA sequence was discovered, and that people called a telomere. And this telomere is at the end of the chromosome, and it's responsible for lifespan of the cell division. So what does it do is when the telomere, once it divides, so of DNA replication, telomere tend to shorten its length. So the cell keep dividing, will keep shortening the telomere length until if it's too short, then the cells cannot divide anymore and die. So that limits the amount of the cell division typically happens in normal cell. So that's why we cannot take the cells out of the living body and make a cell culture for too long time. However, there's an exception, that there's an enzyme called telomerase, which keeps the length of the telomere. That means the cells may be able to divide forever. So the cell become immortal cell line. And The discovery of telomere gave a Nobel Prize in 2009. And why this is such an important discovery, we believe, is over 90% of tumors are known to express telomerase, which is kind of keep the telomere length so that the tumors, the composing tumor cells can keep divide. So if we can target these, we may be able to target the growth of the tumor. So typically the cells are in this G0 status, G0, and that G stands for gap phase. So that is a resting state. However, think about when the cells have to divide and proliferate. Of course, embryonic development, it's a super, very fast growth and proliferation phase. Also, if there's an injury, let's say we have a cut, then that part of the has to have a fill with a cell. So that injury case, even if it's not injury case, a normal tissue function, there's a normal turnover. So think about your skin, you have to take shower, sometimes you have to cleaning. Like every month, your skin cells, that epithelial tissue has to replenish. So that's a normal tissue function, not fast, but it keeps proliferate. Another good example in normal body function is your gut. So from your intestine. So intestines are facing always outside elements, food, and some are probably not very good. So this intestine lining epithelium there's like only several days to keep replenishing themselves. And also our mouth inside the lining, if you have an injury or inside the mouth, you know that they will replace pretty quick. And also very important aspect is immune response. So let's say we've got a viral attack in our body, then our body's immune system recognize that and keeps generating our soldiers, which is specific immune cells against that specific target. So at that time, we need a sudden cell proliferation. So those are like, times when we need the cell to divide and proliferate. So for that, G0 gets into the G1, gap 1. So when does this happen? It's the interaction of the cell with environment or by growth factor, or sometimes it's innate internal mechanism. And that gap 1 prepares the cell to grow for division. And when it goes to the S phase, your DNA is being synthesized. So DNA synthesis, that S coming from this S phase. And afterwards, once the DNA or chromosome, DNA is doubled, you need to also double the cell mass and the organelles of inside the cell. So that's the gap 2 phase. And that prepares for the cell actually divide into 2. So that's called mitosis. I'm talking about somatic cell division, while there is also germ cell division, which is meiosis, which I'm not going to cover it now. So let's quickly review this cell proliferation. So gap 0 is a resting state most of the time, and you get into the cell cycle G1, gap 1, growth and normal metabolic roles until you start to replicate your DNA synthesis. So that's a synthesis S phase, and you have to double your body so they grow and preparing for your mitosis and organelles. So that's a gap 2 phase, and you enter into mitosis phase, and then mitosis phase are divided into 4. I'm not going to get into too much detail, but it's called prophase, metaphase, anaphase, and telophase. So more of this cell cycle is well explained in this YouTube movie, and I encourage you to go and watch this movie. So let's move on to next. So during the embryonic development, the cells, we start from on sperm and egg, and this fertilized egg called as a zygote, and that single cell starts and then form a whole organism and human beings such as, and that requires embryonic development. So that zygote becomes 1 to like 2 to the nth, and they will make a blastocyst. This is a sac with a small inner cell mass. Let me call first the zygote is a diploid cell from 2 haploid gametes. So, and they form develop into blastocyst, which is a mammalian blastula with some differentiation with the inner cell mass. That's called a blastocyst. And cyst means a sac or a bag. And then they will develop into this outer, middle, inner. So you can see outer side ectoderm, external layer, and middle layer, and internal layer. And some cells, of course, will be differentiate into a germ cell, which could become sperm or eggs. So this developmental stage, this outer layer called ectoderm, middle layer called mesoderm, and inner layer called endoderm will differentiate into different types of tissues and cells. So important question to know about this, are all DNA in these different cells, our body, the same or not? And the answer is, of course, our blue point DNAs are the same. However, during this developmental stage, the gene expression, the exquisite control of which genes to turn on and off will decide the fate of these cells. And these specialization we call cell differentiation. So I just want to go through a little bit of a review some terminology on this. So male gamete, the sperm contains chromosome number N, while female gamete called egg will have N. So once fertilized, we will have a zygote, which of course have a 2N. So homologous pairs of chromosomes. So the outermost of layer of cells or tissue in embryo in early development is called, of course, an ectoderm, while middle, meso, within endoderm. And in cell or nucleus having a single set of unpaired chromosome, such as this, that single comes as called haploid. On the other hand, containing 2 complete sets of chromosomes, one from daddy and one from mommy, is called a diploid. And that diploid cell resulting from the fusion of these 2 gametes, which is of course a haploid gamete, will become a diploid, and that is zygote, and or we can call it as a fertilized ovum. And a mature haploid male and female germ cells is called a gamete that's able to unite with another of the opposite sex in sexual reproduction to form a zygote. So next time we'll talk about stem cell, which is more interesting, and cell death. Thank you for your listening.